My name's Luke, although some of you may know me by my middle name Stratus. I'll be discussing antiviral HIV medicines, however I'll be going over reverse transcriptase inhibitors of a non-nucleoside chemical basis. Firstly, I'll speak about the nature of HIV and the development history of non-nucleoside reverse transcriptase inhibitors, followed by HIV's life cycle within the body. Then I'll talk about the drug's mechanism of action, and finally I'll talk about QSAR in regards to NNRTIs. As you may know, HIV is a leading cause of death. The virus attacks CD4 T cells and through this leads to the development of acquired immune deficiency syndrome, also known as AIDS. The earliest clinically reported case was in Norway during 1966. However, the first HIV-1 blood sample was collected in 1959 from a man in Kinshasa, the Democratic Republic of Congo. How he became infected is unknown, but it is commonly believed the virus evolved from the chimpanzee strain and was first acquired through the consumption of chimpanzee flesh. In its structure, we can see an outer envelope and an RNA at its core, with its very own reverse transcriptase enzyme. HIV was published as the cause of AIDS in 1984. The disease is a lentivirus, a subgroup of retroviruses. I will discuss these groups further when I go over the virus's life cycle. Patients with AIDS have heightened susceptibility to both common and opportunistic infections, as well as tumors. HIV has high prevalence. According to the World Health Organization, by the end of 2015, there was 39.8 million people living worldwide with HIV. Here in Australia, 27,000 people are living with HIV. That's only 0.001%. In poorer countries, the virus is much more prevalent, with the highest percentage being in Swaziland at 26%. The virus comes in two forms, HIV-1 and HIV-2. HIV-1 is the more prevalent strain and is responsible for 95% of cases worldwide. The drug category I'll be discussing, NNRTIs, are only effective against HIV-1. As I mentioned previously, HIV is a lentivirus, a type of retrovirus. A retrovirus is an enveloped virus that replicates using host cells through reverse transcription. The subtype lentivirus is characterized by its longer incubation period and their capability of infecting both non-dividing and actively dividing cell types. Regular retroviruses can only infect mitotically active cell types. As it enters, the capsid proteins disperse, and the RNA and reverse transcriptase from the HIV variant are now free in the cell. Reverse transcriptase takes the viral RNA and uses host nucleotides to convert to single-stranded DNA. The enzyme then acts again and converts it to double-stranded DNA. The enzyme integrase transports this DNA into the nucleus of the cell. Within the nucleus, integrase nicks host DNA and allows the HIV DNA to insert itself into the host chromosome. This is what establishes an HIV infection. Messenger RNA then leaves the nucleus to encode for different viral proteins. The protease enzyme digests the viral protein chain into the components, allowing the formation of a mature virion that can go on to infect other CD4 cells. NNRTIs, as the name suggests, reverse transcriptase inhibitors, acts on the reverse transcription step. As the virus approaches the T helper cells, it uses exclusive CD4 T helper cell receptors to bind. This alters conformation of the virion, allowing CCR5, the chemokine co-receptor, to bind. The contents of the envelope protein break through the host cell membrane and fuses the viral and host cell membranes together. It's at this point the viral content enclosed in the capsid floods into the cell, leaving the envelope at the cell's surface. The Riga Institute of Medical Research first discovered NNRTIs when they observed that they non-competitively inhibited the activity of reverse transcriptase in HIV. The NNRTI profile was first defined in 1990. The antiviral family inhibit the enzymes non-competitively and force conformational change in the enzyme, dissolving the enzyme's functionality. As you will see in later slides, the family is very chemically diverse. Drugs with little similarities from a structural standpoint bind to the exact same point on the enzyme. The therapeutic is used amongst a cocktail of others in the highly active antiretroviral therapy program, also known as HART. The program significantly reduces mortality rates of patients and maintains the function of the immune system. This maintenance prevents opportunistic infections from killing patients. As for the mechanism of action, as you can see in Figure 4, each antiviral inhibits a particular step of the HIV life cycle. Fusion and entry inhibitors regulate the virion from merging its genetic content with the T cell. Reverse transcriptase inhibitors, both nucleoside-based and non-nucleoside, stop reverse transcriptase from forming single-stranded and double-stranded DNA from viral RNA. Protease inhibitors act on the enzymes responsible for the cutting of viral proteins, and maturation inhibitors regulate forming of new virion. Reverse transcriptase inhibitors of the nucleoside basis act on the transcription step directly. However, I won't be discussing their mechanism in depth. NNRTIs bind to HIV-1 reverse transcriptase and, as I previously mentioned, cause a conformational change. The enzyme is a heterodimer, and you can think of it as a hand that grasps nucleic acids. The hand is formed by the p66 subunit, made up of 560 amino acids, and can be broken down into the subdomains of fingers and the thumb, as seen in Figure 5. The p51 is the other subunit, and that's comprised of 440 amino acids. If you want more information, I'd suggest searching 1RTH on the PDB. There you'll find an illustration of HIV-1 coupled with nevirapine. The enzyme contains one DNA polymerization active site and one RNase H active site. Both sites reside on the p66 subunit as spatially distinct regions. Interestingly, p51 contains the same amino acid sequence responsible for DNA polymerization. However, it is not functional and is considered to play a large structural role. NNRTIs bind close to but do not overlap the enzyme's active sites. Upon binding, the NNRTI forms a hydrophobic pocket, as can be seen in Figure 5. For more information, please visit Reference 11. Reference 11 yields a study on the crystal structure of HIV-1 reverse transcriptase and the effects NNRTI binding yields upon it. The hydrophobic pocket only exists in the presence of an NNRTI, not in the unbound enzyme. There are both hydrophilic and hydrophobic residues responsible for the binding of NNRTIs. However, the binding pocket is predominantly hydrophobic. For NNRTI effectiveness, three mechanisms are proposed to be responsible. These methods are not exclusive, and all three can occur at the same time. The first mechanism proposes that the pocket forces distortion of the precise geometry of the DNA polymerase catalytic site and deforms the structural elements that form the primer grip. That is, when the hand closes and locks the nucleic acid in for synthesis. The second mechanism theorizes destabilization of the subunits or enhanced subunit interactions. These reactions rely on a delicate balance between both entropy and enthalpy, and any modulation will yield significant repercussions on the enzymatic activity. Thirdly, it is believed that the binding pocket may normally act as a hinge between the fingers and the thumbs, and thereby alterations may slow the thumb subunit, preventing translocation of the subunits and thereby inhibiting the elongation phase of viral DNA. QSAR was difficult with this group of molecules. As you can see in Figure 8, they exhibit large structural differences even though they all bind to the exact same place, obviously with slightly different interactions with the enzyme. However, I did find a study that proved that the log P had a significant effect on the calculation of the IC50 value. This study can be seen in Reference 11. As you can see, increasing the partition coefficient has an exponential effect on the IC50 of these three first-generation FDA-approved non-nucleoside reverse transcriptase inhibitors. That is nevirapine, delaverdine, and efavirenz. As you can see from Figure 9, their structure is very different. That's the end of my presentation. Thank you so much for your time. Does anyone have any questions?